with a big group like this, it might get a little challenging. If you want to raise your hand using the hand icon button, that'll work great. But if it's quiet, feel free to just jump in with a question.
Well, as always, someone has to go first. I'm going to just leap in right now and just say a massive thank you to everyone who's presented today. I've just told my wife, I find it absolutely staggering to think where we were in 2015 and 16, where if we'd had this conference, I think there would have been 1 or 2 people presenting some information. And today, I think it is really tremendous what you guys collectively are achieving. So I just wanted to start by saying thank you very much. It's not a question, it's a thank you. So thank you very much.
Do we have any first questions? If not, I'll throw one out there. All right, no takers to begin with. And feel free to type in chat or just jump in and vocalize as well. We'll be watching chat to ask any questions that show up in chat. So a challenging question here. So we have a big challenge in general in clinical trials and drug development in recruiting patients. When we're dealing with rare cancers, we have a heightening of that challenge in terms of getting enough patients into those trials. I'm staying away from the clinical side of this and getting more to the research side of this. What does the team think about in terms of how we as a community look at some of the drug candidates to elevate them and prioritize them so that the limited set of trial participants that are out there can be gathered around the best candidates, considering that we have so many different options approaching this disease from so many different angles? I know that's a big, broad question, but I'd love to hear your thoughts on, as a community and as a peer reviewing group, how do we evaluate and prioritize candidates?
David, I'm happy to start. I mean, I think, I don't know if we should look at it as limiting or prioritizing, right? I think we should be that lucky to say, you know, we're going to have 5 clinical trials ongoing at any given time. You know, I do think because of the complexity of the clinical trial processes and how to get trials running, we should just push forward on as many fronts as we can. I'm also confident based on what EHE Foundation has done and how it's brought together the community and other organizations in Europe that there'll be a lot of information disseminated to patients, you know, to make sure that they can find the right opportunity. I think more importantly, what I worry about is the clinical trial designs to make sure that we understand who should go on therapies and then how best to figure out response patterns, not only on the clinical trial, but then also correlative work associated with that, right? Because that could be areas where we lose signals or don't appreciate what we see.
I agree with Bill, and in terms of enrollment of patients, I think that could be a little bit different between the US and Europe. Of course, I'm from Europe. In Europe, we have probably less opportunity for patients to enter several studies and to have access to off-label drug, off-label compound. I think that what can be done by the foundation is to make the study known to everybody by having a very good website in which the list of the clinical trial is always updated. On the other side, I absolutely share with Bill, with Dr. Tapp, the importance of designing the right clinical study, not to waste the opportunity of positive results at the first attempt. And in this regard, I think that it's very important to try to define together which is the best way to assess response in epithelioid hemangioma, since RECIST, which are usually applied criteria, could not be the best, especially for the more aggressive variant of the disease for some aspects, but also for the indolent one, since a tumor can grow slowly, but still grow. And finally, I think it's important that you as a community of patients in conjunction with us, tries to explain all the criticisms and the characteristics of the disease to the regulators, since very often they are absolutely not aware of all the challenges that patient and physician are facing to try to get new compound.
And I can add just my 2 cents here. So, I think some of the trials are really exciting, and I think that the foundation does an amazing job of getting that information to patients and being advocates for patients. And access is always going to be the issue, right? So I think on 2 sides. One, as we design the studies, like Dr. Tapp was talking about, thinking about how patients that are maybe geographically distributed or people that are traveling for trials to make their life a little bit easier, to make things more feasible. So making sure that you're, for safety assessments or labs and things like that, like, do you really need the one in the middle of the cycle that's going to make the patient have to come back a second trip? Or can we try to cluster chemotherapy pills, for example, or studies like that, where there might be ultimately some way to do more remote distribution of materials so that patients aren't having to come back and forth necessarily. So these are big problems that are not just for EHE, but things we can continue to work on. And of course, for the foundation, focusing on financial support for patients that are traveling for clinical trials. I get that more than anything from patients is, well, just logistically, I don't know that I'm going to be able to travel for this trial, but if I could, I would.
I will add, I think Silvia said something about indolent versus progressive patients. I mean, and then I saw Bill threw out a comment like maybe we should get a consensus conference together to talk about this. I think that's a great idea, by the way, Bill. But yeah, I think discussions around what types of patients would be best for clinical trials. I mean, when we designed our first trial, we obviously biased it for patients who are progressive, because with the indolent disease, you just don't know if somebody's progressing and if they're indolent, how do you know if they're going to respond without great biomarkers? So I think these are important considerations. I would say the other thing is, now that we actually have some preclinical models that are of the disease, that I would hope that drug companies would come talk to us. They typically do, and get us to evaluate their drugs so they understand from a preclinical level whether they're going to respond or not in bona fide, for instance, EHE cell lines, such that we described in that Silvia's group in Italy has. I think those are important tools, especially when you're talking about a precious resource, which is EHE patients, and we do want to get patients to the most active drugs.
Yeah, I think, and Brian, to kind of build on that, I mean, we've all learned a tremendous deal of how to treat EHE really from our patients and from organizations like this. And there's so many unique variations of what this disease can do to people. And, I'm always struck by how much symptoms play a role in understanding efficacy of agents, where, you may be treating someone and scans look completely stable, but, clearly something's hurting the patient, right? And understanding the patient's voice and how our treatments are helping them or affecting them becomes very important. Now, how we can incorporate that into clinical trials, I think is yet to be determined. But when we speak about what the EHE Foundation's doing, the registry, bringing in patients' voices, how do we include that, in the outcomes of a clinical trial becomes very important in my mind.
Yeah, and I think like the pain syndrome, whatever you want to call it, associated with EHE becomes sort of an interesting consideration. Just even doing a trial to see if you can lessen the pain for those patients, because, I mean, you guys deal with the patients. I don't have to see them in person typically, but I know what a problem that is for them clinically. And even if you can just alleviate pain in some instances, it could be extremely meaningful to a patient. And so I think a conference, Bill, really focused on this topic would be pretty good, and I'd be happy to participate, even though I'm not really a clinician in the sense that you guys are. But I can offer my 2 cents. I think it'd be a good topic.
Yeah.
Yeah, probably, good, especially if you put that in the framework of assessing progression and response, because I think that this is really critical. So symptoms can be an important way to assess if a drug is working or not, but today they by themselves are not very well received. I mean, it's difficult. I remember sirolimus for lymphangioliomyomatosis, in which the drug was approved looking at the improvement of the respiratory functionality, but I think that this is not a very common example among cancers. So if as a community together with patients we can agree upon how we think that response should be assessed, not only by looking at tumor shrinkage, would really be valuable, I think.
I'd just like to maybe throw in one more parameter, and I completely understand that we have to behave in a sensible and safe manner. But when you're considering criteria, please think about time, because for some of our cohort, time is short. And so if you have those meetings, if you could just ask yourselves, can we do this faster? I think that's a great question to just ask occasionally.
Did you mean bring drugs to trial faster, or have this conference faster so we can decide on the criteria?
Both.
All right, Hugh.
But just getting drugs to patients faster could be very fundamental for some patients.
Yeah, I'm sure all the clinicians who prescribe drugs are very frustrated with the pace, of things, but it's difficult to move drugs.
No, absolutely, Brian. We completely understand that, but it's just a great question for us all to ask every so often: is can we do this faster?
Yeah, no, I think it's a good
point. That is not a criticism. Please don't take that as a criticism whatsoever.
I don't,
because I know you guys are as frustrated sometimes as we are at how long things take with regulators and stuff. But
I think you laid down a challenge here. I think a challenge is always really good. A stretch goal. And encouraging us to move faster is always a good thing.
Thank you, Brian.
On the other side, I think that we need to work in parallel, to work out in parallel the two aspects. On one side, the preclinical stuff and new drug development in terms of laboratory stuff is very important. But then when the drug is ready, you need to start this discussion with the regulator. And they are those defining which are the endpoints of the study. So even though the drug is very good, but the endpoint is not the correct one, the study can come out to be negative. And this is very, very frustrating for everybody, because this means that the drug is good only for those patients who enter the study, but not for those who are coming after the end of the study. So to have a position of the community very clear in terms of which are the challenges, what we can do and what cannot be done, and how to assess response, I think that is so important. So it's not wasting time in a sense. It is gaining time, it's gaining a good study, the possibility of a good study.
Well, and I actually think that comes back to a lot of what people were talking about today. The more we know about the biology, the more we have good and accurate models and diverse models, and the better the information, the more likely that the trial can be set up correctly and successful. And I guess from that perspective, after what everyone's heard today, knowing that we're really on the precipice of learning a lot, it sounds like in the upcoming months with a lot of these ongoing projects, does anyone see any gaps in the research that we ought to look at funding or filling or finding teams to look at?
What I think is that linked to the discussion we were having before, to have predictors of response would be great. And in this sense, to continue working together on what can be detected in the blood of our patient related to the tumor in terms of cytokines, of course, but maybe other, things could be very important. And this is something that we may decide to implement a little bit. I mean, it's something that we are already doing together with Paul and with your support, but it's something that could be probably, done even a bit more by collecting blood and investigating what is there in the blood of patients.
And David, I'm not looking at it so much as gaps, because I was utterly impressed with, the breadth and extent of the research that's going on. What I do think, and from Hugh's comment too, is how do we begin to do all of this in parallel, right? We need to really continue to push the research, push the models. We need to push the clinical development of several compounds, right? Not just, one or two, based on your question. We need to be able to get the correlative work from those back into the labs, with those models to understand the response patterns that we're seeing. We need to organize patient groups to make sure we're accruing to trials and treating appropriately. And, then we also need to talk to the regulators, think about the registry, outcome measures, right? So there's a lot of work that needs to be done to say, how do we do this correctly, right? And how do we organize, collaborate? And I think that's a critical question, but also I think could be a very critical opportunity for EHE Foundation to say, what role do we play in that, and how do we really organize this effort?
Yeah, I know one of the factors that I think should be, it's a multifactorial analysis on how to do this best and how to run things in parallel. And I think one of the things we haven't brought in as thoroughly as we could is the regulatory piece. And I think that's something to very aggressively look at. But another one is ultimately, I think, kind of the corporate piece, assuming that early trials might be fundable under grants in some situations, but by the, at the end of the day, some corporate interests will likely have to be involved in drug development. And Jeff, I guess I'd put this question to you. What factors should the researchers be thinking about in terms of information that would be useful to companies in identifying and selecting the right patients and setting up trial conditions? And from a corporate perspective, what concerns do you have about whether certain information is going to be gathered or not, right? At a certain point, there's a cost consideration in how much information you gather on a particular patient, or the criteria by which you set up to get a patient in or not in the trial. Do you have any thoughts about what this team should be thinking about to make sure that the candidates that are rising up to a potential corporate interest at some point are well positioned?
Yeah, I think, so David, I'll try to answer your question as clearly as I can. I think for us, we have a high threshold for bringing new mechanisms into cancer patients, in particular with EHE. And so, for us, we want to have a relatively compelling rationale to bring, it's to make this investment, or it's a gigantic investment for us to bring new molecules into patients. And so, we really want to have a compelling package with regards to the mechanism, the preclinical data supporting the hypothesis. And you can see in the presentation I gave today, we have a little bit of a gap in that, right? And this is part due to the paucity of experimental models over time with EHE, but that's obviously today, there's just a tremendous amount of new models that are being developed. So, and we're, as I mentioned, we're working with some of you already on trying to generate that preclinical proof of concept. And then, safety is a key factor as well. We want to make sure that we have molecules that we bring forth that have a projected reasonable therapeutic index as well. And that's a key factor. Was there another part of the question I missed, David? I apologize.
Oh, I guess there's also an issue associated with, cost. How much, we could spend a lot of money on DNA analysis of each patient to figure out if they're the best candidate or not. What are sort of the corporate?
Critically important. That is not. That's so important to us to really understand, what the different subsets of patients are within a particular disease. So, it obviously is it's critically important to the success. If, we want to be able to define those patients that have the highest probability of responding to our drugs. So the more we really understand and subsetting them with regards to, even today there was some really nice, presentations, trying to understand, downstream of the TAS-CAMP1 fusions, what are the other genetic aberrations that are happening, and which of these are going to be more sensitive to the T transcription factors? Does TAS-CAMP1 potentially bind to other transcription factors and have some non-T dependent activity where it would be essentially refractory to IK930 as an example? So this is just essential to to the way we think about clinical development. And I think it's critical to, number one, the success of the molecule, but more importantly, that we find those patients that are going to benefit the most and don't unnecessarily burden patients that are not going to benefit.
So Jeff, I mean, to that end, I didn't catch all of your talk. I'm so sorry. I had to run off for some operational meeting. But I noticed in your criteria for your study, you didn't say you were going to be doing like correlative studies like NGS to look at something like secondary alterations. Yeah. The other thing I noticed is that you propose to use YAP IHC for qualifying patients. I definitely wouldn't do that. YAP is, I mean, TFE3 IHC is really dangerous. It's present and activated in all sorts of things like angiosarcoma. Campto-1 IHC is good though.
Got it. Okay. Yeah, that's, I mean, so we'll be using these site assays for patient enrollment and confirming that patients have the TAS-CAMT1. So it's really going to be using whatever site assay is used, whether it be an IHC assay, RNA-seq, or FISH assay. And then in terms of kind of what we're intending to do retrospectively, yeah, I mean, that'll be a key part of this, it's a key part of all of our programs, but where I think it's mandatory that, patients have at least an available archive tissue that we can then use for genetic characterization retrospectively, so that we can further learn which ones or define factors that would help us distinguish which patients may be benefiting versus those that are not. And then, we have as a exploratory endpoint in the study, it won't be mandatory for in part because of the specific indications we're going after, but we will be requesting paired tumor biopsies as well. We'll be using those paired biopsies not only for pharmacodynamic purposes, but also for the purpose of trying to understand, additional factors that might be mediating sensitivity to the drug.
Yeah.
So on the other side, I think that, as clinicians, we can surely say that the presence of a fusion is another very, simple element to distinguish patient with a worse outcome, worse expected outcome compared to the other. And this can allow a kind of stratification. I mean, you could even consider something like, after the first 10 patients with or without a fusion, you may want to see which is the kind of response or no response you have, and to decide to move forward with the two types or only one of the two, if responses can be seen only in one or the two subgroups. So we do not know yet which is the biological background for this different presentation, and I think that this should be worked out a lot. This is for for Paul, for Brian, and so on. But from the clinical point of view, we are very clear signs of worse outcome that can be included in the study designed from the beginning.
We've got about 7 minutes left for the roundtable, and a comment and a question came in, so let me throw those out. The comment noted that part of the delay that we should look to avoid is lawyers in the process. Things like CDAs and MTAs getting signed. I actually have, being a lawyer, I have some clever thoughts on how EHE Foundation and others might be able to assist with that, but we'll take that offline. Thank you for the point. That is actually worth considering as a way to speed up the process. And then we've got a toss-up question for the group from Guy Weinberg. I have preliminary data suggesting plasma-soluble endoglin as a potential biomarker. How would we leverage the Biobank and EHE Foundation contacts to do a robust test? This could serve as an example for how to use the system to test a hypothesis. Any comments from the group on that?
So I can comment on the, and I guess there's a session tomorrow on the UK Biobank that we've set up. And part of that is collecting blood specimens as well. And we have a mechanism by which you can apply for access to the blood specimens, and it will go through a project review panel. But yes, so I think the process is starting. Whether you get enough numbers to show a firm finding. That's a separate question. But yeah, so that there are avenues to potentially get access to bloods and others. So.
Yeah, Paul, I would agree. And I would also say, like, when we think about prospective design of clinical trials, how to collect serial blood and at times also where we can correlate potential changes in something that you're looking at with response patterns and symptoms, right? So that becomes really important as well, in addition to the biobank. Yeah,
and I would just say, I guess, from a bench scientist point of view, whenever trials are being planned, it's very important. It's a missed opportunity if we're not collecting specimens of blood to learn from our patients. So I would definitely put some thought into some of the correlated aspects, especially with such a ultra rare entity.
Yeah, and I mean, just to build off of that too, and if the clinical trialist is not talking to the lab, right, they may not be collecting the right sample at the right time under the right conditions, and then that's also a way. So that level of communication is critical.
We've got a question coming in from John Lamar. Please go ahead, John.
Yeah, so I mean, I think the biobank is that we've been hearing about is a great idea, but I'm wondering if we can do more to get more clinicians to be aware of this biobank, because right now it's either driven by the centers that are part of these studies or the patients themselves, and there are a lot of hospitals that are going to see EHE patients and do biopsies and then not have collected the right data at the right time. So is it possible to start reaching out to other surgeons that might see EHEs but not be quite aware of our of this community, and start engaging with them so that they know this biobank is out there? Is there a way the EHE Foundation can do that? So for example, at my hospital, they see a couple every couple of years, and those samples are just going to waste because they don't see a benefit in doing anything with them. They get one or two at a time and they're not going to bank them. So there's probably a lot of that in the smaller institutions. And can we capitalize on those samples to make sure that every single EHE sample is being used for some of the things we've been talking about?
And I see Denise's hand up that may be coming in with an answer to that question. Denise?
Yeah, hi everybody. Yeah, so great question, John. And so we've just gotten the biobank really settled at the Cleveland Clinic Biorepository this year. We started off last year, took a little bit of a left turn, got settled down. So this year's initiative on biobank is to do exactly what you're talking about and really reach out to those centers, and we've already started doing that on a minor scale, but that's the initiative to do this year, is to really work with med-onc, surgical oncologists, making sure that we're getting access to those patients early and often. We're making good progress on consenting patients, but we've got to go at it from both angles, just like you're talking about.
Maybe just to follow, do you have a good program or sort of IRB approval set up that you could share with other institutions so that they can maybe help in identifying the types of tissues and things from their institutions? Because that's a big part of it, especially for smaller institutions. Getting the IRB set up for these samples is very tedious, and if you don't know what you're doing, it can be very challenging. So another thing EHE Foundation could do is sort of formalize that process so that we make sure we're capturing everything we need from all these patients.
Yeah, and we have central IRB approval on the protocol. We've not actually hit any roadblocks from an IRB perspective. We've had a couple of stumbling blocks with institutions who are a little more rigorous on whether or not they'd like to allow samples to go out of their institutions. We've worked around that in most instances, all the way through the patient walking in with the box themselves and then getting it FedExed out of the institution. But generally speaking, with that central IRB approved protocol and coming in and letting the surgical team know that this is the patient's wish, we're not getting any pushback.
And this works out perfectly as the allotted time for wrapping up the roundtable and going to co-chair closing comments by Dr. Tapp. Thank you everyone for attending, and for those you who will be on tomorrow as well, I'll look forward to seeing you if you're not going to be there tomorrow. Again, thank you much for showing up. Great to see so many people out on looking at this topic.
Yeah, and David, thank you. I just wanted to thank you too, because you do an absolutely wonderful job moderating all of these sessions. So thank you for that. And I appreciate just taking a few seconds to thank EHE Foundation and all of the presenters. First of all, I'd like to absolutely congratulate all of the presenters, because the work that we saw here is just dramatic and remarkable. And I think you said it, 5 years ago, if you think back with our knowledge of this disease, and where we are then and where we are today in our understanding of EHE and the signaling pathways is just dramatic. And not only then, but to hear today with the development of models, I know in listening to all the talks today that it is not easy, it is not a quick thing, but congratulations to just your passion and how stubbornly and fastidious you're continuing to pursue these models. I think it's going to have tremendous dividends. And then as we're seeing that, we already have the reporting of SARC-33, which is trametinib in EHE, which is remarkable to have the first study solely in this disease, and what we are going to learn from that, not only in design, but also in outcomes. And then to have the prospect of new compounds, pharma involvement coming into EHE, and then the thought of some of the drug discovery that we're beginning to see. So this is extremely hopeful. And I want to kind of shift the focus back a little bit to the EHE Foundation, because so much of this work is galvanized by what you've been able to put together, just the organization, the philanthropy, and importantly, your involvement in helping us understand the disease. And I think that's really important. And as we've always spoken, understanding the disease clinically has been really tough, and having a patient's voice at the table to talk about what they go through has really shaped what we in the clinical community have been able to accomplish. And it's allowed for large consensus meetings, white papers, really thoughts on how to approach and study this disease. And kind of the challenge that I'll put forward to the next year is exactly what I was speaking about in the roundtable, which is, further organization of our community, inclusive to EHE Foundation and patient advocacy, to say, how do we now begin to move this work forward rapidly on many different fronts? And I think this is where we really need to get granular. We need to have an understanding of clinical development of compounds, of clinical trials, discussions with regulatory bodies, correlative research that comes out of the clinical work as we continue to really see the development of this wonderful science that's going on. And this is the type of partnership that we need in the academic side. And so I just want to thank EHE Foundation as well, and really look forward to kind of pushing the envelope for the next year with everyone who's on this call. So thank you very much. And I will pass the torch over to Jenni, who I believe will have the closing remarks.
Yes, thank you so much, Dr. Tapp. I appreciate it. Okay, so I have been asked to wrap up the day. If you missed my intro this morning, my name is Jenni Kovach. I'm the president of the EHE Foundation, and I'm also a patient myself. So to say that I am sitting here humbled, amazed, and appreciative of all of you. Thank you for giving your time and attention to EHE. I think I can speak for the EHE community as a whole when we just say, thank you. We appreciate you. We are your partner. I have a bunch of notes from today and takeaways of ways that we can engage, we can continue to try and move the needle, but we're here as your partner to raise money, to raise awareness, to continue high levels of engagement from EHE patients. So with that being said, I just want to say thank you to a couple of people, a couple of companies. First, I'm going to start with our sponsors, our lead sponsor, Ikena Oncology. Thank you very much. Our other sponsors are Every Life Foundation and the Cleveland Clinic Sarcoma Program. Then I also just want to do another call out to Dr. Rubin and Dr. Stacchiotti and Dr. Tapp for co-chairing the conference. We appreciate you guys and giving your time and helping make today possible. If you missed any part of today, it has been recorded and it will be available on the event site, I think for the next 90 days to registered participants. And then if you aren't scheduled tomorrow, please consider joining us. It is our first ever patient conference, which begins at 10 a.m. Eastern Standard Time. We are so excited. So I think what I should say at this point is with that, I think it's a wrap. And I appreciate everybody. Thank you very much.